So hello everyone. So this session we are going to deal with study plan. So another important aspect of your entire behavioral setup is that you need to have the study plan. Very important that this sort of sequence is followed. So you need to plan the entire experiment before you start, because you need some time for training, figure out how many sessions that is required, and then you will start conducting the pre-stroke test. If it is Parkinsonian model, then pre-surgical test has to be performed, which will serve as the control. Alright, if there are no other control groups, this itself can be used as a control to compare the performance before and after the creation of stroke. That is very important. Then you induce the lesion. Alright. So once the induced lesion, there has to be a recovery period. Only then you can start doing the test again. Alright. So this is allowed for brain swelling that can happen after inducing the stroke, or for that matter the wound healing, the consciousness that needs to be recovered completely. All that healing and recovery should happen before you subject the animal for the test. Let us say if there are some electrode implantation planned, then again you need to have a period of recovery, then you are going to start acquiring the signals. Or if you are going to have a drug delivery, then you should have a recovery, then you start looking at the test again. Alright. So such study plan, and then what is the performance that you are going to look at, all that has to be ready before you start the experiment. Alright. So briefly about the Visha test apparatus, I am not covering the lever press apparatus, because we are trying to look at the motor cortex, finer movement of the forelimb as an example of the stroke model. Once you create the stroke model, the recovery is pretty much good for the proximal muscles. The distal muscle usually suffers, and that is the theory in humans. Alright. Whereas even in rodents we have observed that the proximal recovery does happen. That is because of the redundancy built in the sensory nervous system. So usually it is the caudal forelimb area which will take some time, or you want to really restrict your study to the caudal forelimb, where you are going to look at only the digit movement or the forepaw movement occurring, you are not really interested in the shoulder and elbow movement of the animal, then this is the best apparatus to look at. Alright. So how is it that you are going to plan it? So this apparatus has a slit. Alright. So these are the dimensions there, totally about 36 centimeter in height. Alright. So you will have a shelf in front of that slit, which is around 4 centimeter from the anterior wall of this chamber, or the front portion of the chamber. There is a slit. From the slit there is a platform or the plate where there are two wells in it, where the food will be kept, and that is where the rat is going to grab the food pellet from. So you need to understand the forelimb has to go through this slit, reach the well and grab the pellet, take the limb back in, then feed the pellet itself. So this is the entire sequence of movement that needs to happen. So that is the beauty of this apparatus. It is a pretty simple apparatus, but we can record pretty complex movement that is happening once a rat is adequately trained. And this is the entire setup that you need to have, but you can modify this particular test apparatus as per the requirement of your own study. For example, if there is a electrical signal acquisition happening, then this entire thing can be covered in what is known as Faraday cage. So this again we have covered. So basically to isolate the test apparatus from the electrical noise that can happen in your area. alright. Then you need to plan your video apparatus. So that's the top camera from bottom, from front and from the side, so that the forelimb movement can be analyzed and you can do the kinematic analysis if you have recorded, and there are various softwares that are available to study the movements, alright. So there is no end to it. You can make things as objectively as possible. You can use a lot of complex software that are available, but the whole idea is that you are studying the movements objectively, alright, and you are reducing the subjective variation that can happen with just visual inspection. That is the whole idea. And if there are electrophysiological study that you have planned, then it is all the more important to study these movements and then correlate with the neural signals that you are recording, alright. That is where these kind of video recording, kinematic analysis plays a huge role if you are if you are trying to decode the neural signal which is responsible for that particular movement that you are trying to look at. So that is how it is important. Based on your objectives, you can add n number of additional accessories into this particular apparatus and then study it. And there are various modifications that can happen with this apparatus as well. So this is the example as to how exactly the rat performs this particular single pellet retrieval task. You need to remember single pellet, that is this food pellet retrieval task, alright. So that is the task that is going to be performed by the rat when it is adequately trained, alright. How we train it will come into the next session. In the next few slides we will come to that. So this is some example which I have shown how the rat is going to insert its forepaw and then grab it. If you see it, it does look like the human grab, alright. So that is where the beauty lies. You are trying to look at what may be happening in the human brain by studying the rodent brain. But then you need to sort of replicate that sort of movement in a rodent. This is the apparatus which is close to that, alright. So you can see how the hand is being brought, and long even has this very distinct digit, lot of this space in between the digit, and you see the phalangeal joints which are pretty long. That is another additional advantage of long even rat models, alright. So just to sort of give you an overview of this test, basically it brings the forelimb through the slit and it grabs it, retrieves it, and then takes it inside and feeds itself, alright. So that is the overview. So you are trying to look at really that part of the test apparatus. So obviously your camera will, this is the recording from the front camera, this is a snapshot from the front camera. So similarly you need to have a side camera. These two cameras are very, at the bare minimum that you need to have to analyze the movement. If there is a luxury of having another one below and on top, you can further analyze it. Every single movement can be analyzed further, alright. So coming to the most important aspect, you are dealing with a small animal, alright. So where you cannot really give any verbal instructions. So training becomes a very important aspect if you are trying to use the animal model to replicate any sort of disease process, and if you want to study the behavior, training it plays very important role, and that is something which is going to consume a lot of time as well. So but then there is a learning curve. Once you gone through it, the time gets shorter. But then the most important thing is, how are you going to deprive the food? What are the steps, precautions that you need to take when you are subjecting the animal for food deprivation? Does it really fall within the ethical ambit of the animal ethical committee? All that is something you really need to consider before preparing the protocol for that particular experiment, alright. So I have taken an example of this Visha test where the animal has to grab the food pellet, and it will be easy to notice that a well-fed animal will never try to put its hand into that slot and grab the animal. It really needs to be hungry and motivated and trained and should, and trained to locate this food pellet, to understand where the slit is. All that takes some time, and so the training takes a very important role to come across that sort of hurdles. Alright, so in next few slides we will see how we will train the rat or mice for this particular experiment. So food deprivation is the first and foremost step within the ethical criteria. You need to satisfy the ethical guidelines for food deprivation, which is very, very important, for a simple reason that the underfed rat is not going to perform the experiment altogether, or if the well fed rat may not even attempt the test. So you need to strike a balance between this well fed and underfed rat. It shouldn't lose its body weight, it shouldn't lose the performance activity because it is not fed and because it is really tired. Alright, so that is a balance you need to strike. So for that, general recommendation is that around 3 days of food deprivation is necessary to start training, and then it can continue as long as you are going to train it. Alright, so you need to weigh each mouse and obtain the baseline body weight before the food deprivation starts. That is very important, that the body weight loss should not decrease more than 10% per day. Alright, so that is important of establishing a baseline body weight, so that as the experiment progresses, as the training proceeds, you need to make sure that the rat does not lose 10% per day body weight, because if it loses more than that, definitely the animal is going to underperform. Alright, so classically before 3 days prior to the pre-training, you are going to start the animal on standard lab diet. So there are again a number of diets that are available. If you are trying to do the single pellet study, the best diet that need to be arranged is precision food pellet. Alright, so that is something you need to procure. They are available in West easily, but in India to arrange this particular precision food pellet has to go through a lot of these license issues, and it has to be imported from various food suppliers, which is a round pellet which comes within the grasp of the animal, within the forepaw of the animal, and it can nicely wrap the, if you can see this picture, this is the precision food pellet. The idea is that the shape is uniform, so that the movement and the behavior learning that can happen is smooth. Alright, whereas if you are trying to make small pieces of the regular food chunks that are usually fed, that's going to vary in shape and size. So the rat will take a longer time, and it may not even like it, and it may not make an attempt. It would rather probably would starve. Alright, so but then even if when it starve, it might grabs it, but it becomes hyperactive, which also is something we need to avoid. It shouldn't be hyperactive, it shouldn't be fidgety, then the drop rates are going to be more, the failure rates are going to be more. So to make sure all that is taken care of, the recommended food is the precision food pellets. Alright, that is the whole idea, to have this precision food pellet, and you need to feed 0.05 gram of food per gram body weight per day. That is the formula you need to calculate, and give it for at least 12 hours, and then the regular feed. Water should be unlimited supply. Alright, so general recommendation, rat weighing 200 grams, you can give 10 grams of food. So once you subject the rat to this particular formula, this is what you need to ensure, that it body weight does not drop more than 10% per day. So before you subject the animal for this particular test, the animal should be left in the training box without the food pellet to acquaint to the testing environment. That is also very, very important. Alright, So it's going to stiff around, it's going to explode the atmosphere, and if it continues that, continue to do that during your test period, you're going to lose out on number of days, and it probably even lose the body weight because you started your food deprivation. So it's very important that the animal sort of gets acquainted with the test apparatus. So there is something known as training box before you actually start the Visha test. That's a Visha test apparatus where there is only one single slit. So training box, it will have multiple slits like that, and outside there is a platform with all the amount of food that the rat can have. So it gets acquainted by grabbing the food through the slits that are there in the food cage itself, and get familiarized with the food retrieval through the slit. So that is one form of training apparatus before you actually put into the test apparatus. Alright. So if you do not have the training box, you can even directly start training in the training apparatus itself, but it may take a longer time for the rat to learn that particular movement. And you need to perform this the food deprivation and the test apparatus for at least 10 to 15 minutes daily for 3 days. This is kind of pre-training where you leave the rat in the training box with around 20 pellets in this platform, and the rat grabs through it. So if you do that 10 to 15 minutes daily for around 3 days along with food deprivation, then your rat is ready to take on the test apparatus itself. So next 3 to 5 days, first 3 days you look into food deprivation. Next from 3 to 5 days you do what is known as shaping. Okay, that is to determine the dominance, forelimb dominance. So as I said, in the animal, even in animals like humans, there are dominants. You are a right-handed person or you are a left-handed person, whereas in animal you do have some dominance where animal will use its preferred limb. Alright, it might use the right forelimb or it might use the left forelimb. So you need to determine what sort of dominance that your subject has. So that shaping is very important, because if you are trying to induce stroke for a right-handed animal, you need to do it on the left hemisphere. Excuse me, so that the right forelimb becomes paralyzed, and then you can do various implantations or studies from there onwards. So you need to determine the dominance. So the next 3 to 5 days you need to place the single slit side of the training chamber facing downwards to see if it uses its dominant limb. Alright. Then press the foot tray up against the front wall of the training chamber to allow the seats accessible to the mouse. So this is kind of direct feeding. Alright. You are directly trying to feed the animal by getting the food pellet right into the slit, and see which is the hand that is used by the animal to grab the food pellet when it is easily available. So then you need to do it for 20 reaching attempts within 20 minutes. Okay. Then you have attained your shaping target, where either it will be using right forelimb or it will be using left forelimb. Alright. So whichever limb it uses for more than 70% reaches, that is the dominant limb for that particular animal. Alright. So that is how the shaping is done. Next comes the actual training, which will go on for 7 days, where the actual steps, actual movements are captured. Alright. So then you are going to put the individual seeds or food pellet in the food platform in the divot that you have. There are 2 food wells in front of the slit that you have. Alright. So you will be keeping the food pellet in that, and then you will look at the preferred paw of the mouse. If it is right hand mouse, use the mouse's right side. Alright. So that is very, very important to look at. Once those movements are happening, you need to classify into these 3 attempts, whether it is successful or is it going to drop it, or is there a failure. That is very important. The success is considered when the preferred paw is used, it grasps the pellet, retrieves the seed and feeds it into mouth. That entire step is important, only then you will be considering it as a success and you note it as success. Whereas it uses the paw and grasp it, but it drops it before it takes it in the mouth, then that has to be considered drop. When it either misses the seed or knocks it off completely from the holding plate, then it is considered as failure. So you need to do this multiple times and give multiple trials for 30 attempts, for at least 20 minutes, whichever comes first per day. That you need to give a trial of SDF. Keep marking it and see how it's going to perform. All right, and then only the mice is brought back into the home cage after training, and then you need to give the daily food quantity that the rat is supposed to take. All right, so that's very important. So we will go through this Visha test in detail in the next session, but to summarize, basically you need to give it a scoring as per the success rate. Normal is going to be definitely 0. You are trying to look at the impairment in these particular reaching. Once the training is done, once the success rate is good, then you are going to rate the movements that is happening in the rat forelimb according with this 10 steps that are happening when the rat grabs the pellet and feeds itself. These are the movement sequence that are happening. So, and then you are going to score it appropriately. So, if it is only mildly impaired, you can give the score of 0.5. If it is totally impaired, then it is 1. So, usually the hemiplegic rat after the stroke, the score will be close to 10 or somewhere at least 8 to 10, alright, usually. So, obviously this scoring will help you to understand how the rat is recovering from the stroke. The recovery can happen because of the plasticity, because of the collateral supply, it does recover. So, then you should have a very objective scale to look at the impairment and the recovery rate. Alright. So, with that we will close today's session. In the next session we will try and complete the various Visha test scores, how we can look into each and every movement that is happening, and then we will try and look at what are the improvements that can be brought upon, what sort of sequences can be automated. So this is sort of really laborious, and it is too demanding in terms of time and efforts to look at each and every single step manually. So there are these lot of these automation that have come into it, wherein you can deliver the food pellet automatically, you can record the sequence of movements automatically, then you can bring in AI algorithms to do the kinematic analysis. So next session we will deal with it, and after that we can start looking into the Parkinsonian models. Alright, thank you for your kind attention.